Hello, ladies and gentlemen. Welcome back to Chapter 12, where we are talking about prokaryotes. This is the third section on prokaryotes, where we are primarily going to be talking about how bacteria are able to reproduce and change their genetic material, which kind of counts as reproduction. Okay, first off, a review we mentioned before about binary fission, which is the most common type of reproduction. You start out with a single bacteria. It is asexual. You only need to have one bacteria. The DNA, which is in a ring, reproduces, and you get a second copy. And then you have cytokinesis, where the cell splits, and you end up with two identical daughter cells. This happens over and over again, and you simply get a lot of the same type of cell. However, there is no genetic change. It is simply multiplication of that type of cell. Now, the second type of situation we're going to talk about is a little strange. It's called transduction, and this is where you actually have genetic change of bacteria because of a virus attacking them. You see this little creature up at the top that looks kind of like a robot spider? That is known as a bacteriophage, or phage for short. Basically, they are viruses that specifically attack bacteria. Now, you can see primarily that what's happening is the DNA from the phage is entering into the bacteria. You can see that viruses are a lot smaller than bacteria, by the way. And that DNA from the virus enters. Now, what ends up happening is the virus starts multiplying and multiplying and multiplying inside of the bacteria. And what can happen is little pieces of the virus or of the DNA of the bacteria can enter into the virus. And you can see that right here. There's a virus that actually has taken a piece of the bacteria DNA, not just the virus DNA. So although it's a virus, it has bacteria DNA inside of it, unlike the others, which have the normal virus DNA. Then what happens is lots of times the bacteria are destroyed, not always, but the viruses then get released. Whether they get destroyed or not, the viruses leave. And if that strange virus that has the bacterial DNA goes to attack another bacteria, it's not going to create a virus inside, but what happens is the DNA from the other bacteria actually will join the DNA of that bacteria, or possibly have it become a plasmid. So what ends up happening is you actually have a genetic change of the bacteria, and that can completely transform the bacteria depending on what part of the DNA it got from the other bacteria. So this is one of the ways that bacteria can radically change quickly, because then if you have this altered bacteria go through binary fission, it would just reproduce and reproduce. And this is sometimes how we have problems when dealing with trying to get rid of certain bacteria, is they change and they make it so it's unable to do whatever we did before to get rid of it. Okay. The third technique of DNA change in bacteria is called bacterial conjugation. Okay, on the left you can see that you have the normal nucleoid with a chromosome, and you have a plasmid. Now, this bacteria on the left is different from the one on the right. This one doesn't have a plasmid. So what ends up happening is this bacteria, the donor, actually creates a structure called a pilus, which is like a little tube, and the plasmid breaks apart and replicates and goes through that plasmid, or pilus, into the recipient bacteria. And you end up with this new genetic information that's entering in, and it can completely change what that bacteria is like. And it'll change the structures and things of that bacteria, and you end up with a new type of bacteria. And that is conjugation. And it's worth noting that the bacteria DNA is only going in one direction. It's not being exchanged back and forth. You simply have the DNA from the plasmid of one go into the other. Okay, another type of DNA change can happen due to bacterial transformation. This is a little strange, but basically if you have bacteria and surrounding them is DNA from another bacteria, say one that exploded or some strange guy in a laboratory, the bacteria will actually absorb in the DNA from the outside. Even if it's not from bacteria, even if it's just a gene from something. They did an experiment where it was the gene for jellyfish glowing. So the bacteria took in this DNA, and you have two different options. One, it completely incorporates it, which is how the bacteria ended up glowing, or you end up with that additional bacteria or additional DNA becoming a plasmid, which can change the structures and things within the bacteria and making it function differently. Okay, just in case I haven't explained thoroughly, you have a short video to have somebody else explain it also.
People commonly understand how major plants and animals exchange genetic information, but the ways in which microbes like bacteria do something similar are often overlooked. Bacteria rely on several different mechanisms for the exchange of genetic materials. In one case, a bacterial cell may transfer genetic information directly into another bacterial cell through a process called conjugation. In conjugation, one bacterium grows a duct, called a pilus, which attaches to the other bacterium. A genetic element, known as a plasmid, is then passed through the pilus from the donor cell to the recipient. In another case, viruses play a role in genetic exchange between bacteria. Bacterial viruses, or bacteriophages, sometimes just called phages, ordinarily attach themselves to bacterial cells and then inject their genetic material into the cells. Such viruses hijack bacteria, using bacterial cell components to generate new phage particles. In some cases, a phage's reproduction cycle kills the host bacterium. In other cases, the bacterium survives. This occurs when the virus's DNA becomes incorporated into the bacterium's DNA. At this stage, the virus depends on the host bacterium for the replication of new phage particles. When the new phages emerge from the host, some may carry pieces of bacterial DNA. If the new phages attach to other bacteria and the DNA becomes incorporated into bacterial DNA, new genetic types of bacteria may be produced. This process, in which bacteriophages assist in the genetic recombination of bacteria, is known as transduction.